Next, we will discuss diagnosis and therapy of genetic diseases. For diagnosis, we'll talk about gene mapping and linkage analysis, as well as molecular methods with and without embryo selection. And for gene therapy, we'll talk about gene replacement therapy and gene blocking therapy. So for gene mapping, it's important to discuss polymorphic markers and make sure you understand what these are. There's different types of markers that we can use to map the genome out to look for similarities and differences, to look for identical loci, and different types of polymorphisms that are seen in order to identify things, for example, for paternity tests and things of that nature. So let's go through a few of these. One of them is restriction fragment length polymorphisms, or RFLPs. These are variations in nucleotide sequences that can modify the presence of restriction endonuclease sites from person to person. So remember, endonuclease sites are looking for specific sequences, specific patterns within the genome, and depending on where you have these polymorphic patterns, where you have these endonuclease sites, you can have different fragment lengths from person to person. So using those fragment lengths, you can actually identify somebody's DNA. So those are RFLPs. Something similar, variable number of tandem repeats, or VNTRs. This is varying number of repeating units in a specific region of a chromosome. So instead of looking at endonuclease sites, they look at repeating units. Not very often used, but still something that you want to know. A couple of other polymorphic markers, short tandem repeat polymorphisms, STRPs, also known as microsatellites. These are similar to VNTRs, usually about 2 to 6 bases long. So what you do here with these is you can use PCR and amplify them, then digest by restriction enzymes, leading to, similar to the RFLPs, leading to varying fragment lengths between individuals. This is the testing, this microsatellite testing is what they use now for paternity testing and in forensic cases. Another type of polymorphic marker is the SNP, or single nucleotide polymorphisms, positions where only 2 nucleotide possibilities exist, for example, either an A or a T, and that's the only thing that can be there, and that can be identified by DNA sequencing. Now, moving on, let's talk about linkage analysis. Linkage analysis is used to determine chromosome location of a gene, so where on a chromosome is a specific gene. Now, how do we do this? Let's go back to meiosis. Remember that during meiosis there's crossover events that can occur between homologous chromosomes. Now, because they can cross over, you can get switching of genes even if they're on the same chromosome. Now, when we go through meiosis, remember that genes will assort, independent assortment and segregation. Now, if they're not on the same chromosome, they will segregate independently, and they're said to be unlinked. Genes on different chromosomes can randomly assort, independently assort, and they're said to be unlinked. If genes are on the same chromosome, what we'll see is when we have random assignment or random independent assortment, those genes will not segregate independently, and if that's the case, then they're said to be linked. So here's a picture of what we're talking about using this idea of linkage analysis. So we see in A, we have 4 different genes, G1, G2, M1, M2, located on 4 different chromosomes. Now those are randomly going to assort, just depending on chance. So if a cell gets G1, then 50% of the time it will get M1, and 50% of the time it will get M2, just by random chance. And those genes, G and M, would be said to be unlinked in that case. Now let's look at section B, or possibility B. Let's say that they are on the same chromosome. You see, G1 and M1 are on one chromosome, and G2 and M2 are on another chromosome. So in this case, they're far apart on that chromosome. And remember what we said, that we get crossing over during meiosis. So these homologous chromosomes will come together, and they can cross over. And they cross over frequently enough so that if a cell gets G1, half the time it'll get M1, and half the time it'll get M2. So if they're far enough apart, they would appear to be unlinked, because they independently assort in the same numbers and in the same probabilities that if they weren't on the same chromosome. Now, look at C, situation C. We have G and M very close together. G1 and M1 very close together on one chromosome, and G2 and M2 very close together on another chromosome. Now, when these homologous chromosomes come together during meiosis, even with this crossing over that occurs, those genes are so close together that it's not likely that crossing over events are going to occur in between them, so that whenever you get G1, you're more likely to get M1 as well, and you're not likely to see G1 with M2. And if, in those situations, when that arises, you would say the G1 and M1 are linked. That's what we call linkage. Now we can use this linkage to discover recombination frequencies, or we can use recombination frequencies to discover how close together two genes are on a chromosome, and we can use that to map chromosomes. Now, again, the closer two genes are on the chromosome, the smaller the frequency of a recombination, the less likely they are to have that crossover event between them. 1 centimorgan, you might hear the term centimorgan when we talk about recombinations and gene mapping. Well, 1 centimorgan means that there's a 1% frequency of recombination between two genes. Okay? Now, LOD scores is another thing that might come up. LOD stands for log of the odds, and this is estimating probability of linkage in big populations. A lot of times when you're looking at chromosome maps of a certain family, there's not enough to be able to say whether two chromosomes are linked or not. There's just not enough samples. But if you bring multiple families together, for example, that have the same type of disease and want to map those chromosomes, then you can use LOD scores to try to estimate the probability of linkage. Now, we're not going to go into all of the math that encompasses, because it's way beyond the scope of the boards, but what you do need to know is that if the LOD is greater than 3, that indicates that there's linkage present. If the LOD is less than -2, that means there's no linkage. Now, you'll see something called theta. Highest theta is the most likely recombination frequency. So that's what you need to know about centimorgans and LODs. Other than that, it's a lot of math that you really don't need to have the specifics for in order to do well on your boards. All right, let's switch gears and get into diagnostics. Genetic diagnostics uses molecular techniques to directly identify disease-causing mutations in the individuals being tested. Tests are very accurate, provided the molecular nature of the disease in the population has been identified. If we know the gene that we're looking for, and better yet, if we know the mutation in the gene that we're looking for, then we can be very accurate in our diagnosis because of this molecular nature. Techniques that are most commonly used for diagnosis are things like PCR amplicon sequencing, probe hybridization, and restriction digestion. Diagnostics can be applied to early embryos after an IVF procedure to select those embryos without a mutation for implantation. So again, in that diagram, when we're talking about somebody that may have a genetic disease that doesn't want to pass that on to their kids but want to have children, so they choose to have in vitro fertilization. Well, you can take gametes from the partners, you can fertilize them in vitro, and then you can take those early embryos, you can apply these diagnostic tests to them and see if they have the mutation or not, right? Now, they can also be done once a woman is pregnant on chorionic villus sampling or amniocentesis aspirates. So it just depends on the person whether or not they want it before pregnancy or after pregnancy. So what can we look at? Amniocentesis, performed at 14 weeks, can detect chromosomal anomalies. We can look for trisomies and things like that. We can also look at alpha-fetoprotein levels, do an AFP assay. Now, increased assay levels, increased alpha-fetoprotein levels, either in the amniotic fluid and in the maternal serum, those are indicative of neural tube defects. Now, if those alpha-fetoprotein levels are decreased, generously decreased, then that's usually indicative of Down syndrome. The other thing we can look at is chorionic villus sampling, so we can do a trophoblast biopsy. We can do that a little bit earlier than amniocentesis, about the 9th week, so we can find out if there's a genetic problem earlier on, about 5 weeks earlier. The downside to that is that they may give a false positive. So, a couple of therapies that are in the works for genetics. This is kind of an up-and-coming field, a new field, now that we have all of the molecular techniques that we do. One of these therapies that is being investigated right now is the transgene therapy. So transgenic therapy uses a vector, usually a virus, to add exogenous DNA in to replace a patient's gene. Retroviral vectors are good candidates for this approach since a normal part of their replication is to integrate into human genomic DNA. Remember that retroviruses have reverse transcriptase, and they can become DNA, and they can integrate their DNA, their viral genomic DNA, into the human genome. Now, they've tried this to treat SCID, specific types of SCID, and it's been used successfully. So that's the plus side to it. The downside to it, the problem that they're still trying to develop, is the fact that these viral vectors can go awry, and they can cause mutations on integration leading to cancer. So if they happen to splice into a part of the genome that has a predisposition for cancer development, well, then we have a problem. We can't really control where these viruses are inserting themselves into the genome. The other downside to this transgene therapy is it will only allow treatment of recessive or loss-of-function diseases unless an endogenous sequence can be targeted. All we can do is gene replacement, right? If you're not making a protein, we can insert the gene to make that protein. Now, gene blocking therapy is kind of the reverse of that. Here, they're working on strategies to develop therapies for gain-of-function mutations. So the idea behind it is that we can prevent the expression of aberrant gene products. One strategy is the use of antisense RNA, or RNAi, which can lead to destruction of the mRNA transcript of mutated genes. So in this case, what we would be doing is looking to knock down transcription and translation of gain-of-function mutations. Now, this has been successfully used in lab animals and human cell lines, but hasn't been translated all the way to humans yet. So let's go through a couple of more random pedigrees here just for practice. Here, what do you think of this one? We've got a male in one generation passing it to daughters and not sons in the second generation. And then looking at the third generation, the daughters can pass it to either females or males. So if that's the case, then what kind of pattern are we looking at? Fathers passing it to daughters and not to sons. So this would probably be an X-linked, right? And it's in every generation. You get mixing when the females have it, so you would say this would be an X-linked dominant type of inheritance pattern. Here's another inheritance pattern for you. We have a random person with the disease here, so this is one of those questions, where the heck does this disease come from? Nobody else is sick. What you have to look for, and the dead giveaway here, is that consanguineous relationship, that double horizontal line. When you see that, you should always think autosomal recessive. That concludes this section of the lecture, and also the online genetics course.